Hyphal tips may be the places where data streams together to determine the speed and direction of growth. But how do tips in one part of the network know what tips are doing in other, more distant parts of the network? We stumble back into Olson's conundrum. His bioluminescent Pinellas cultures were able to coordinate their behavior over time periods too short to be caused by chemicals moving from A to B through the network. The mycelium of some fungal species grow into fairy rings that stretch across hundreds of meters, reaching hundreds of years in age, and then somehow produce a single circle of mushrooms in a synchronized flush. In Body's experiments with foraging mycelium, only one part of the network discovered the new block of wood, but the behavior of the entire mycelium changed, and changed rapidly. How are mycelial networks able to communicate with themselves? How does information travel across mycelial networks so quickly? There are a number of possibilities. Some researchers suggest that mycelial networks might transmit developmental cues using changes in pressure or flow, because mycelium is a continuous hydraulic network like a car's braking system. A sudden change in pressure in one part could, in principle, be felt rapidly everywhere else. Some have observed that metabolic activity, such as the accumulation and release of compounds within hyphal compartments, can take place in regular pulses that could help the synchronized behavior across a network. Olson, for his part, turned his attention to one of the few other options that remained: electricity. It has long been known that animals use electrical impulses, or action potentials, to communicate between different parts of their bodies. Neurons, the long, electrically excitable nerve cells that coordinate animal behavior, have their own field of study, neuroscience. Although electrical signaling is normally thought of as an animal talent, animals aren't alone in producing action potentials. Plants and algae produce them, and it has been known since the 1970s that some types of fungi do also. Bacteria, too, are electrically excitable. Cable bacteria form long, electrically conductive filaments known as nanowires. And it has been known since 2015 that bacterial colonies can coordinate their activity using action potential like waves of electrical activity. Nonetheless, few mycologists imagined that it could play an important role in fungal lives. In the mid-1990s, in Olson's department at Lund University in Sweden, there was a research group working on insect neurobiology. In their experiments, they measured the activity of neurons by inserting fine glass microelectrodes into moth brains. Olson approached them and asked if he could use their rig to ask a simple question. What would happen if he replaced the moth brains with fungal mycelium? The neuroscientists were intrigued. In principle, fungal hyphae could be well adapted to conduct electrical impulses. They are coated with proteins that insulate them, which would allow waves of electrical activity to travel long distances without dissipating. Animal nerve cells have an analogous insulating sheath. Moreover, the cells in a mycelium are continuous with one another, possibly allowing impulses initiated in one part of the network to reach another part without interruption. Olson chose the species of fungus carefully. He surmised that if electrical communication systems did exist in fungi, it would be easier to detect in species with a greater need for communication over long distances. Just to be safe, he chose a honey fungus, or Armorillia, the species that forms the record-holding mycelial networks that stretch over kilometers and reach thousands of years in age. When Olson inserted the microelectrodes into Amaryllia's hyphal strands, he detected regular action potentials, impulses, firing at a rate very close to that of animals' sensory neurons, around 4 impulses per second, which traveled along hyphae at a speed of at least 0.5 millimeter per second, some 10 times faster than the fastest rate of fluid flow measured in a fungal hyphae. This caught his attention, but in itself it didn't suggest that the impulses formed the basis of a rapid signaling system. Electrical activity can only play a role in fungal communication if it is sensitive to stimulation. Olson decided to measure the response of the fungus to blocks of wood, which is food for this species. Olson set up the rig and placed a block of wood onto the mycelium several centimeters from the electrodes. What he found was extraordinary. When the wood came into contact with the mycelium, the firing rate of the impulses doubled. When he removed the block of wood, the firing rate returned to normal. To make sure that the fungi weren't just responding to the weight of the wooden block, he placed an inedible plastic block of the same size and weight onto the mycelium. The fungus didn't respond. Olson went on to test a range of other species of fungus, including a mycorrhizal fungus growing on the root system of a plant, pleurotus, or oyster mushroom mycelium, and serpula, the dry rot fungus found fruiting in the oven at Haddon Hill. They all generated action potentials, like impulses, and were sensitive to a range of different stimuli. Olson hypothesized that electrical signaling was a realistic way for a wide variety of fungi to send messages between different parts of themselves, messages that conveyed information about food sources, injury, local conditions within the fungus, or the presence of other individuals around it. Many of the neurobiologists Olson was working with became excited that mycelial networks could behave like brains. It was the first reaction from all the insect people, Olson recalled. They were thinking of these big mycelial networks in the forest sending electrical signals around themselves. They imagined that maybe they were just big brains lying there. I admit that I hadn't been able to ignore the superficial resemblance either. Olson's findings suggested that mycelium might form fantastically complex networks of electrically excitable cells. Brains, too, are fantastically complex networks of electrically excitable cells. I don't think they're brains, Olson explained to me. I had to hold back the brain concept. As soon as one says it, people start thinking of brains like ours, where we have language and process thoughts to make decisions. His caution is well placed. Brain is a trigger word burdened with the concepts that spend most of their time in the animal world. When we say brain, Olson continued, all associations are with animal brains. Besides, as he pointed out, brains behave like brains because of the way they're built. The architecture of animal brains is very different from that of a fungal network. In animal brains, neurons connect with other neurons at junctions called synapses. At synapses, signals can combine with other signals. Neurotransmitter molecules pass along synapses and allow different neurons to behave in different ways. Some excite other neurons, some inhibit them. Mycelial networks don't share any of these features. But if fungi did use waves of electrical activity to transmit signals around a network, wouldn't we think of mycelium as at least a brain-like phenomenon? In Olson's view, there could be other ways to regulate electrical impulses in mycelial networks to create brain-like circuits, gates, and oscillators. In some fungi, hyphae are divided into compartments by pores, which can be sensitively regulated. Opening or closing a pore changes the strength of the signal that passes from one compartment to another, whether chemical, pressure, or electrical. If sudden changes in the electrical charge within a high full compartment could open or close a pore, Olson mused, a burst of impulses could change the way subsequent signals passed along the hypha and form a simple learning loop. What's more, hyphae branch. If two impulses converge on one spot, they would both influence pore conductivity, integrating signals from different branches. You do not need much knowledge of how computers work to realize that such systems can create decision gates, Olson told me. If you combine these systems in a flexible and adaptable network, we have the possibility for a brain that could learn and remember. He held the word brain at a safe distance, clamped in the forceps of quotation marks to emphasize that a metaphor was in play. That fungi could use electrical signaling as a basis for rapid communication has not been lost on Andrew Adamatsky, the director of the Unconventional Computing Laboratory. In 2018, he inserted electrodes into whole oyster mushrooms sprouting in clusters from blocks of mycelium and detected spontaneous waves of electrical activity. When he held a flame up to mushrooms, different mushrooms within the cluster responded with a sharp electrical spike. Shortly afterward, he published a paper called Towards Fungal Computer. In it, he proposed that mycelial networks compute information encoded in spikes of electrical activity. If we knew how a mycelial network would respond to given stimulus, Adamatsky argues, we could treat it like a living circuit board. By stimulating the mycelium, for example, using a flame or a chemical, we could input data into the fungal computer. A fungal computer may sound fantastical, but biocomputing is a fastly growing field. Adamatsky has spent years developing ways to use slime molds as sensors in computers. These prototype biocomputers use slime molds to solve a range of geometrical problems. The slime mold networks can be modified, for instance by cutting a connection, to alter the set of logical functions implemented by the network. Adamatsky's idea of a fungal computer is just an application of slime mold computing to another type of network-based organism. As Adamatsky observes, the mycelial networks of some species of fungus are more convenient for computing than slime molds. They form longer-lived networks and don't morph into new shapes quite so quickly. They are also larger, with more junctions between hyphae. It is at these junctions, what Olson describes as decision gates and what Adamatsky describes as elementary processors, that signals from different branches of the network would interact and combine. Adamatsky estimates that a network of honey fungus stretched over 15 hectares would have nearly 1 trillion such processing units. For Adamatsky, the point of fungal computers is not to replace silicon chips. Fungal reactions are too slow for that. Rather, he thinks humans could use mycelium growing in an ecosystem as a large-scale environmental sensor. Fungal networks, he reasons, are monitoring a large number of data streams as part of their everyday existence. If we could plug into mycelial networks and interpret the signals they use to process information, we could learn more about what was happening in an ecosystem. Fungi could report changes in soil quality, water purity, pollution, or any other features of the environment in which they are sensitive to. We're some way off. Computing with living network-based organisms is in its infancy, and many questions remain unanswered. Olson and Adamatsky have shown that mycelium can be electrically sensitive, but they haven't shown that electrical impulses can link a stimulus to a response. It is as if you have struck a pin into your toe, detected the nerve impulse that traveled through your body, but hadn't been able to measure your reaction to the pain. This is a challenge for the future. In the 23 years between Olson's study on mycelium and Adamatsky's study on oyster mushrooms, no further research was conducted on electrical signaling in fungi. If he had the resources to pursue the line of inquiry, Olson told me, he would try to demonstrate a clear psychological response to changes in electrical activity and decode the pattern of electrical impulses. His dream is to hook up a fungus with a computer and communicate with it, to use electrical signals to get the fungus to change its behavior. All sorts of weird and wonderful experiments could be done if this turns out to be right.